Hi, I'm Sun Sui-Ning from School of Biosciences. The presentation topic today will be tissue engineering, growing muscle cells in a petri dish. Generally, it's called in vitro meat. The reasons for why in vitro meat is introduced is because over the next few decades, the world population will increase from 7 billion to 9 billion people. Coupled with the skyrocketing development of the countries such as China and India, this population growth will place new burdens on the world's food supply, especially the production of meat. The world cannot sustain an increased burden on its natural resources due to heightened demand for meat. Tissue engineering and cloning are already being implemented to help ease some of the burdens on the world food supply. So, what does it mean by in vitro meat? In vitro meat is a method of producing meat where the protein cells are grown from a culture of animal stem cells in a laboratory. As a single cell could multiply rapidly, the meat produces enough to feed the global population for a year. After the cells are multiplied, they are attached to a scaffold and soaked with nutrients. They may also be mechanically stretched to increase their size and protein content. The resulting cells can then be harvested, seasoned, cooked, and consumed as a boneless processed meat, such as sausage, hamburger, or chicken nuggets. Most of the people think that clone meat and in vitro meat are the same, but in fact, they are not. Cloning involves somatic cell nuclear transfer technique, which creates a viable embryo from somatic cell and an enucleated egg cell. The embryo form is then used to implant livestock such as cow or sheep, when then give birth and are raised in the traditional fashion. However, in vitro meat only involves replication of cells in culture, not cloning of whole animals. Scientists have associated the development of in vitro meat to a number of advantages. The first important advantage of producing in vitro meat is better control over meat composition and quality by manipulating the flavor, fatty acid composition, fat content, and ratio of polyunsaturated fatty acids through composition of the cultured medium. For example, omega-3 fatty acids can be added to in vitro meat as a health bonus. Hence, the chances of getting blocked arteries can be reduced. Secondly, due to strict quality control rules, the chances of meat contamination and incidence of food-borne diseases could be significantly reduced. In addition, the risk of exposure to pesticides, arsenic, dioxins, and hormones associated with conventional meat could also be significantly reduced. Third advantage may be the prevention of global warming. A study by researchers at Oxford and the University of Amsterdam found that in vitro meat was much more efficient and environmental friendly, generating only 4% greenhouse gas emissions, reducing the energy needs of meat generation by up to 45%, and requiring only 2% of the land that the global meat or livestock industry does. This is in contrast to cattle farming, responsible for 18% of greenhouse gases and causing more damage to the environment than the combined effects of the world's transportation system. Besides these advantages, there are some disadvantages that need to take into consideration too. The public will be concerned about the unnaturalness of in vitro meat and possible unknown long-term risks to health due to the addition of growth hormones. Currently, because of the small scale of production, culturing in vitro meat is very expensive. Keeping the cells warm, healthy, well-fed, and free of contamination takes incredible labor, energy, and a huge amount of investment. The production of in vitro meat starts with extracting muscle stem cells from animals, such as cows, chickens, via biopsy. Biodegradable or edible scaffolds are provided in order to produce 3D in vitro meat. Muscle cells are grown in three-dimensional bits that stretch with small changes in temperature or pH. Then, the scaffolds containing the cells are placed in a bioreactor together with a growth medium. The isolated stem cells are then developed into muscle cells. The cultivated muscle cells will bulk up into solid muscle fibers. To do so, they are affixed to a soluble polymeric sugar scaffold and trained by building tension between two anchor points in the bioreactor. Soluble polymer duct systems are used to allow medium to flow through the large slices of meat. Finally, the edible ultra-thin sheets of muscle cells would be vacuum-packed and sent to a meat processing facility to make a ground meat product, such as hamburger. Natural consistency of meat in terms of protein composition, fat, tissue, and etc. is recreated to make the tissue edible, taste, and have a right texture. So, in summary, in vitro meat offers health and environmental advantages over existing meat production system. However, due to the extremely expensive and may bring adverse health consequences, the meat has a long way to go before it can be cheaply and safely made.